Hello, this is Scott Carpenter, and tonight I'm going to go on a different subject than the Sasquatch Awareness Project. I promise you a summary, and the final chapters are coming up, but I'm very excited. I want to talk to you tonight about the Sasquatch DNA study, or the Sasquatch genome study. And Dr. Ketchum has been starting to do interviews again with the help of the Club of No Return and encouragement from Steve Ishdahl. The Club of No Return banded together, and hundreds if not thousands of people donated to get Dr. Ketchum's GoFundMe over the top, what she needed to complete the genome study, and from where it left off as far as continuing and giving further analysis, and so they could finish a paper, another paper on the study with more details. And she also has dogman samples, and this will allow her to complete the work on those dogman samples. And so she's kind of ramping back up, which I'm glad to see. Bless her heart. She's, kind of had to lay low and do the best she could, and it's I'm very happy for her, and it's good to see her back. And I just wanted to talk a little bit tonight. Spoiler alert, I don't know, I've watched one of her podcasts, so if you haven't watched any of her podcasts, you may wait till after. But, talking about, the podcast, unfortunately, it was just for her, it was just voice only. And there wasn't, the one I saw, there wasn't any anything going on as far as let me pull that up while I'm thinking about it. There wasn't anything as far as results, like she didn't have any presentations or anything. And so what I decided to do, I've written more than one book. I mean, the Nephilim Among Us has the results in it by permission. The same thing with Truth Denied. The Truth Denied, and I'm sure y'all are tired of these shameless plugs, but this, the Truth Denied is the story written from my point of view, as far as what was going on around me as a submitter of hair samples, DNA samples, and working with David Pilates and the North American Bigfoot Search Group. And it's the study, what happened around the study. The truth was denied, and it's the whole sordid story from start to finish about what happened with the study, and how we were all attacked. Especially Dr. Ketchum was savagely attacked, and her livelihood was taken away from her, all because she dared to go after the truth. And this is definitely vindication for her, and I am so happy to see it. But I wanted to go over real quick, because on her interviews, and I don't know, I'm hopeful that on later interviews she'll have some presentations and some stuff where she can get into it, and she referenced people to the Sasquatch genome study page, which she has, and I'll put in the links here. But I just, in chapter 10 of The Truth Denied, I went over the results of the Genome Project, and I just wanted to, what's on the screen, I just wanted to scan through that real quick. It's not going to be like a deep dive, but it's just going to let people know. I think sometimes people don't realize how serious this study was. That there was a DNA project team, and there were multiple disciplines on this study. Dr. Ketchum was the the face of it and the lead of it and the head scientist, but she had a team, and she was the one that was out front and taking the slings and arrows. But a lot of people just think she just made this up in a back room somewhere, no, she didn't. She worked with a team. And I just wanted to go over that real quick to give people background, and I'm sure Dr. Ketchum will probably get into it more, but I want to support her, and I feel like people need to know. And I've got several emails already, and a bunch of instant messages or from Messenger asking me about the study again, and hey, weren't you involved with her, and what were the results, and oh, I didn't realize this and I didn't realize that. So kind of refresh everyone's memory. So as we're looking here on the let me make sure it's recording. It's recording, sorry. So you have to excuse me, I don't have a soundproof studio. I have an office and a bunch of wild animals in the neighborhood. So like I said, all the sources here, you can see here, all the sources for this chapter can be found at the Sasquatch Genome Project's official website, which is the SasquatchGenomeProject.org. And this was a team. And, let me read this first part. There is a common misconception that Dr. Ketchum worked alone with little or no assistance. This is not true. Dr. Ketchum collaborated with at least 10 other individuals with expertise in many areas, to include but not limited to forensics, DNA analysis, microbiology, molecular biology, biophysics, and biochemistry. The following is a list of the team, their credentials, and their contribution to the study. And I'm just going to scroll slowly, and you can look at them. These are the different doctors that worked on the study and what they did, and a little bit about their background. So these people were not, fly-by-nighters. These weren't, these were published professionals in their fields, experts in their field. And I'm going to scroll here. And of course it's in the book, shameless plug again, blah, blah, blah. So there's kind of, who analyzed data, who wrote and edited the manuscript, who did the electron microscopy, it's a hard word to say, analyzed and wrote the hair analysis, that sort of thing. They used David Spence, who was he was the trace evidence supervisor with the Southwestern Institute of Forensic Scientists Criminal Investigations Laboratory at Dallas County, Texas. He's no slouch. So anyway, just kind of go through, and then there were multiple laboratories used. That's another thing that people need to understand, that Dr. Ketchum was very smart, and she knew that if she did, after doing a couple, three, and getting results, she knew she had to do blind studies and get them out. So they hired laboratories from all over the country, and they would just send them the DNA and say, do the results, and send us. And so it was a true blind study. This is a list of the different laboratories. You had North Louisiana Criminalistics Laboratory, DNA Diagnostics, that was her lab. Southwestern Institute of Forensic Science, Dallas, Texas. Family Tree DNA in Houston, Texas. SeqRite of Houston. Helix Biological Laboratory, Department of Biological Sciences, Wayne State University, Detroit, Michigan. USC, Los Angeles. University of Texas at Arlington. UNT Center for Human Identification, University of North Texas Health and Science Center, Fort Worth, Texas A&M, College Station, University of North Carolina, Chapel Hill, Texas Veterinary Medical Diagnostics Labs, Texas A&M University College Station, Hughley Pathology Consultants, Fort Worth, Texas. So these different laboratories were not fly-by-nights. You know, they weren't. You know, these were mainline institutions. So I'm going to kind of skip through to get down to the brass tacks of all this. I don't want to bore you with all the details. Just letting you know that you know, this is all as I'm scrolling, all the different things. But I wanted to get down to real quick, since DNA. What is DNA? How does it work? I want to do this real quick. I had a little section called DNA Boot Camp. And DNA is short for deoxyribonucleic acid. It is a molecule that contains the genetic code for all organisms. DNA in each cell of an organism. DNA is in each cell of an organism and instructs the cells what proteins to make. The cell's proteins determines its function. DNA is inherited by the children from their parents. The DNA in a person is a combination of the DNA from each of their parents. It's a double helix, and I go explain that. There are two types. Here's what's important. There are two types of DNA, nuclear and mitochondrial. The mitochondrial DNA is found in the cell mitochondria and is from the mother only. The nuclear DNA is found in the cell nucleus and is a combination of the genetic code from both the father and the mother. Mitochondrial DNA is more abundant and easier to extract and test. Nuclear DNA is more difficult to extract and test. Therefore, samples in quality and quantity are required for successful extraction of nuclear DNA. Kind of here's the little nice little internet thing I found, you know, kind of showing you where the DNA comes from. So another thing that you're going to hear a lot about is called GenBank and BLAST. GenBank is an institution that collects and stores DNA sequences for all life forms on this planet. Once a DNA sequence is placed into the database, comparative analysis can be run on it using a special software program called BLAST. It's like a search engine. BLAST is a software engine that compares a DNA sequence against all the DNA sequences stored in the GenBank database and reports any matches from this database. At the time I wrote this book, when was that? Not 2019, 2019, 20, 18, can't even remember. But at that time there were 152,599,200, oh, I'm sorry, 152,599,230,112 samples in the database. I think I did that right. So this is a quick thing of how it's formed. I won't bore you with that. Like I said, so here are the testing and the results. And I'm kind of going to go over this real quick so you can understand. There were a total of 111 samples in this study. Hair, saliva, blood, enamel were in the study. I think one of the big things that I've highlighted here was that, you know, they screened the hair and they knew what a Sasquatch hair looked like under the microscope. They knew its characteristics. And one of the very important things, and you need to remember this, is a non-human hair, in other words, it was not human, it was not animal, yielded human mitochondrial DNA. That's important. Here I show the difference. You can see here, this is a Sasquatch hair. and here, and this is a human hair. Blew that up. Below is the summary of, here's the 111 samples of blood, tissue, and hair and other types of specimens were studied, characterized and hypothesized to be obtained from the elusive hominids in the North American, commonly referred to as Sasquatch. This is from the study. The DNA was extracted, purified, and a subset of these samples that survived regular screening for wildlife species identification, mitochondrial DNA sequencing, specific genetic loci sequencing, forensic short tandem repeat STR testing, whole genome single nucleotide polymorphism SNP bead array analysis, and next generation whole genome sequencing were conducted on purported Sasquatch DNA samples gathered from various locations in North America. Additionally, histopathologic and electron microscopic examination were performed on a large tissue sample. In summary, our data indicate that the Sasquatch has a human mitochondrial DNA but possesses nuclear DNA that is a structural mosaic consisting of a human and a novel non-human DNA. So, let me get down here. I want to get down scrolling fast because I got all the different, this is detailed. Electron. So, because I go in and I explain everything. So here is the Sasquatch Genome Project conclusion summary. And this is my words, you know, kind of giving you an overview of what went on. So, we have over a 5-year period, more than 100 samples of hair, tissue, tree bark shavings, saliva, flesh, teeth, fingernails, tooth enamel, and blood were submitted by a nationwide team of individuals from 14 U.S. states and 2 Canadian provinces. All samples were screened prior to DNA analysis to eliminate common wildlife species using a variety of methodologies, including microscopic, morphological examination, and a comparison against a large reference collection of known American wildlife hair. In other words, they were compared to everything, and the hair was compared to everything they had. Dog, cat, everything. Over 100 hair samples passed the initial screen and were determined not to be human or from a known wildlife species. It was discovered during the testing, this is important, that Sasquatch hair alone would not yield mitochondrial DNA. Normally, human hair, dog hair, cat hair, horse hair, you can, just from a hair strand, you can get mitochondrial DNA. But with a Sasquatch hair, it would not, their hair alone, a strand, would not yield DNA. This was unusual because both human and animal hair will yield mitochondrial DNA. Only hair samples with ample skin tags were selected for the study. In other words, the hair had to have a skin tag on it. To avoid contamination, the hair samples were forensically processed and cleaned. Let me read that one more time for all the nut jobs that run around. It was contamination, blah, blah, blah. To avoid contamination, the hair samples were forensically processed and cleaned. The hair samples were sent to multiple labs for blind testing. All samples were determined to be human, with the result consistent with human mitochondrial DNA. Non-human hair yielded human mitochondrial DNA. And there it is. Results from the purported Sasquatch samples tested over the previous 2 decades, that's previous stuff, had always yielded the results of human mitochondrial DNA. In other words, before the study, you know, David Pilates had a real good article talking about this on his website. Basically, just over and over again in the 80s and 90s and in the 2000s, people would collect, they'd see a Sasquatch and they'd collect the hair and they'd send it off for testing. And of course, they would only do the mitochondrial side, so since they had hair, it's all they could get, and it would come back human, and they would declare, oh, it must have been contaminated. And, you know, over and over and over and over. They never considered once, hey, maybe this thing is a hybrid, or maybe it's human. Maybe, this is correct. So this kind of, this verified, the actual separate results and this result verified the fact that, they verified each other. Results from purported Sasquatch samples tested over the previous 2 decades had always yielded a result of human mitochondrial DNA. These results were always dismissed as human contamination. The results of the Sasquatch DNA project support the conclusion that these samples were, in fact, Sasquatch samples. 30 mitochondrial DNA samples were selected for complete genome sequencing and HV region haplotype screening results. The haplotypes will show you what region of the country the DNA came from. The kind of gives you the origin, where the originating area locale. Sorry, I'm goofing that up. But anyway, the samples yielded haplotypes that showed the origination of the Sasquatches to be from Asia, Africa, and Europe. In other words, they didn't, a lot of people thought, oh, well, there'll be a lot of Native American or whatever, and they'll be from North America. And they weren't. Their haplotype showed them coming from Asia, Africa, Europe, actually I should have put in there the Middle East as well. Only 4 out of the 30 haplotypes originated close to where the samples were collected in North America. Also, none of the samples contained mitochondrial DNA from apes, Neanderthals, or Denisovians. God, it's a hard word to say. Let me say it again. They were not apes, they were not Neanderthal, and they are not related to Denisovians. They did some gene sequencing, the amylogenin gene, excuse me, I messed that one up, amylogenin gene, from the nuclear DNA of the same 30 samples was sequenced to determine the sex of the subject. This gene tells you what sex it is. Mixed results were obtained for human chromosomes, with some failing to amplify while others would only amplify the Y chromosome. These were pure non-contaminated samples that yielded human mitochondrial DNA, but when they ran the nuclear DNA, the DNA that comes from the father, to see if they could figure out what sex they were, it either failed to amplify when they used human primers. In other words, this is proving that the sample, was a human hybrid. The nuclear DNA was not human. You had human mitochondrial DNA. So logic says if I test the nuclear side with human primers, I should get, the results. They didn't. They basically either failed or they would only amplify the Y chromosome. So, failed at times to amplify when human primers are used for sex determination. It's important. The samples were tested for the MC1R human and Neanderthal red hair color gene, MYH16. So the samples were tested for the MC1R human and Neanderthal red hair color gene, MYH16. The gene is associated with the sagittal-crested apes and was analyzed in an effort to determine if the unknown hominid was related to apes, and the TAP1 human antigen gene associated with breast cancer, diabetes, and several other syndromes. The MCR1 results were human, no ape, or Neanderthal sequences. The MYH16 results were human. The TAP1 results showed unknown when compared to the GenBank database. But the samples matched each other. In other words, this TAP1 gene that they looked for didn't match anything. It was the human antigen gene associated with breast cancer, diabetes, and several other syndromes. The TAP1 didn't match anything in the database, human or animal, but the results matched each other, saying basically this is the species and they're related to each other. That's important. This is stuff that scientists overlooked or misled people about You know, so you've got this unknown human hybrid that while there's no there's no nothing in the database for this gene, for this sequence, they do match each other. 24 samples were tested on the whole human genome, 2.5 million SNPs, Illumina bead array platform using an Illumina iScan instrument. That's a very complicated instrument to look at the different samples and yield results. All the 24 samples failed to meet the human threshold of 95% SNP performance. In other words, the samples were human mitochondrial DNA, hybrid nuclear, and when they ran them through this on this bead array platform, they did not yield results that were human. It takes 95% on the SNP performance to say you're human. The results range from 53% to 89% on the performance. Attempts were made to duplicate these results with degraded human control. Melba talked about this. And the attempts were unsuccessful. The degraded human scored 97. In other words, when they took human blood and degraded it and ran it through, still came out 97. And so even pure, pristine DNA samples from the Sasquatch would not test human for on this bead array on the SNPs. The mitochondrial for these samples tested human, but the nuclear results for the SNPs were non-human. So here again we have human mitochondrial, non-human nuclear. A core sample was taken from flash sample number 26 and examined under electron microscopes. The results showed the nuclear DNA to be very unusual and non-human. Electron micrographs of the DNA revealed unusual double-strand, single-strand, double-strand transitions. In other words, the human genome, or our DNA is a double-strand helix. When they looked at a living creature and they looked at his nuclear DNA under the electron microscope, it had double-strand, single-strand, double-strand. Shouldn't be alive. This thing shouldn't even be breathing. Yet here it was, and this, of course, this is evidence that suggests that the Sasquatch nuclear DNA had some time in the past been genetically manipulated. Three samples contained ample nuclear DNA were selected for the next generation whole genome sequencing. In other words, they were able to get a complete genome, 3 terabytes worth of data. The DNA from these 3 samples were sequenced using the next generation Illumina platform at the University of Texas Southwestern in Dallas, Texas. In-depth analysis of all 3 genomic sequences, and these are the samples, 26, 31, and 140, was performed at the University of Texas Southwestern, and alignment confirmed by the university. In other words, they were, 2 different labs looked at them and confirmed the alignment, and confirmed by the University of North Texas Health Science Center. The resulting genomes were compared to the complete GenBank database using the BLAST search. In other words, so they took these sequences, took the nuclear DNA, they got the sequences using this sequencer, and they got good, pristine, clear DNA samples or DNA sequences. University of Texas Southwestern got it out, and then the University of North Texas Health Science Center, checked the work, made sure that, they didn't get it wrong. And so you had 2 different organizations, 2 different places say, hey, this is right. The genomes did not match any organism in the GenBank database. The 3 genomes were a completely unknown species. Yet, the 3 samples aligned and matched each other. Hear me talk about that before? So, while there was nothing in the GenBank of all the different species, humans, animals, it didn't match anything, but they did match each other, showing that they are, of course, a species, and they're related to each other. In other words, these 3 samples came from the same species. Here's the summation. If I can read this without collapsing here or getting tongue-tied. DNA from over 100 separate samples of hair, tissue, toenail, bark scraping, saliva, and blood obtained from dozens of collection sites throughout North America have been extracted, analyzed, and sequenced. The hair samples were not consistent with human or any known wildlife hairs. Analysis of the DNA found that the mitochondrial DNA was human, but the nuclear DNA was from an unknown source. All known ape and relic hominid species, such as Neanderthal and Denisovan, were eliminated as being contributors to both the nuclear and mitochondrial sequences. Whole genome analysis showed that the Sasquatch is an unknown human hybrid species with some DNA sequences not matching humans or any known species. And then this is from the DNA, this is from the Sasquatch Genome Project. This is their conclusion. And of course there's the source, you can go there. But this is what they summed up. Sorry about that. It kicked me or went over time here. I'm still recording. Yeah, sorry. I'm still learning my equipment. So, all right. So from the Sasquatch Genome Project, here is that summary. The data clearly supports that these hominids exist as a novel species of primate. Novel means unknown or unknown to science. So the data further suggests that they are human hybrids originating from human females. This hybridization can be likened to humans with the hybridization can be likened to humans with Denisovan admixture resulting from Denisovan males mating with human females. This is referencing another study where they found that human females had also interbred with Denisovans. The same type of mating potential occurred with the Sasquatch. However, in the case of the Sasquatch, the admixture is human. So, and so through preliminary analysis, though preliminary analysis supports the hybridization hypothesis, alternately, it could also be hypothesized that the Sasquatch are human in origin, having been isolated in close breeding populations for thousands of years. Nevertheless, they gave two hypotheses here of how, what kind of hybridization it could be. The data conclusively proves that the Sasquatch exist and as an extant hominid and are a direct maternal descent of modern humans. So basically that was the conclusion of the paper. And like I said, the biggest takeaway is they're human hybrids. Their mitochondrial DNA is human female. The nuclear is unknown and not human. And like I said, the non-human hair yielded human nuclear DNA. So I hope that was helpful. I've got a lot of questions, and this is wholly in support of Melba and what she's doing. And a lot of people, like I said, I've got, I don't know, probably a dozen emails about it today, and probably twice that in messages on Messenger. So this, instead of writing 50 replies to emails, here you go. And I think this will help kind of get people who are trying to figure out, okay, what's going on with Dr. Ketchum. You kind of mentioned her before, you wrote about her before, and now she's back on the scene, and she's telling us this is kind of a primer to get everybody up to speed to where we're at now. And moving forward, she's going to continue to give, she has more analysis to do, pie charts and stuff, actually showing the percentages of what a Sasquatch is, like the human and the other things that it is. And then, of course, she's got the Dogman DNA. And she's going to be, it's human. It has a human mitochondrial DNA. All these hybrids, all these cryptids seem to, when you start building a hybrid or a cryptid, you start with human female, and then you start manipulating. And I know that, I've talked about it before, the Dogman has canine nuclear DNA and other things, which I'm sure she'll when she puts that information out here hopefully soon. We'll all get to see all this, and I think it'll be great when we're getting the truth out. So with that being said, I encourage y'all to continue to watch Melba. I'm posting links as they come up on my Facebook feed, and please watch. And if you can contribute, go to her GoFundMe webpage or site and contribute. Guys, just keep hers in prayers. Usually, the trolls and the attack squad, I expect to come out. I'm sure this will activate those people, and unfortunately, here they come. And it's to be expected. When you get over the target, they're gonna begin shooting at you. And hopefully, this will help cut off some of that at the knees and give you the true information and the truth about the study, so you'll understand what's going on. So when people start lying, and they will, and they are, when they start lying and putting out misinformation, you can discern that and just push it to the side. So with that being said, guys, I hope you enjoyed this little primer on the Sasquatch DNA study. I called it the Sasquatch DNA study because most people knew it as that. It's officially the Sasquatch Genome Project. And guys, just take care out there. I'll be back soon with some more content. I got some fun content to show you. I might got a capture I kind of came upon from when it was back trail from the dog man. It's not a dog man, but it's a funky looking thing. And on the next video I'll do that, and then I'll get back into the Sasquatch Awareness Project. So guys, God bless, take care, and we'll talk to you soon.